Hi guys. So today in class we did a strawberry DNA lab. So I am actually going to perform the lab for you guys right now so you can visualize what happened. So every person got a strawberry. I'm going to do two so we have plenty of DNA to see. Because we're doing two, we're going to need two sets of buffer. So this is a soap, water, and salt mixture measured to 10 milliliters, a coffee filter, a coffee stirrer that is wooden, and a beaker, which is the clinking you can hear in the video. So the first thing I'm going to do is I'm actually going to put these strawberries in the same bag. So strawberries in the same bag. We don't need this one, so we can put it off to the side. We're going to seal our bag. We're going to squish our strawberries. Just really crush them up. Put it down. And what we're doing here is we are exposing the cell membrane. We're really breaking into that cell membrane to make it so we can use our strawberry. Once it's at about this consistency, I'm going to go ahead and add both of our buffer solutions. I'm going to do so carefully as to not make the soap bubble. 2. Okay. We're going to reseal this and we're going to continue to mix our strawberry for just a few minutes, a few moments to really get that soap in. The soap is actually breaking down the lipid membrane of the strawberry. And it's not only the cell membrane, but we're also breaking down the lipid membrane around the nucleus. The salt is going to attract itself to the DNA. This is going to help the DNA get itself out of the cell membrane. So we're pretty, crushed up here, broken down. You can see that we're very liquidy, which is what we want. So now I'm going to filter. The filtering process is going to remove any of the chunks of the strawberry from the juice. Because of the salt in the soap, our DNA should be out of the nucleus by now, that we are actually sifting the DNA through our coffee filter. So let me show you what I'm doing here. So I have this, and what I'm going to do is just very carefully filter this into my beaker. So my filter's falling, and it fell totally in, which is not what we want. So let me go grab a new one. So because I'm actually working with two strawberries, I grabbed a bigger beaker and a funnel for this to work. So I'm going to take this coffee filter out. I'm going to place this coffee filter in our beaker, or in our funnel. This is going to allow us to really see what is going on in our beaker. So what we have happening is that separation of the juice from the strawberry and the chunks from our strawberry. I'm going to let this run for about 15 minutes and replay the video when we get back. All right, we're about 5 minutes into our wait time. We're already getting a good amount of strawberry liquid with some DNA. I'm going to let it sit for about 5 more minutes and come back with some ice cold ethyl alcohol, which is going to bring the DNA to the surface, and we'll be able to scoop it out. All right, so it's been a few more minutes, and it doesn't look like I have much more juice to come separate out from our solid, and I have the ethyl alcohol. So what I'm going to do is I'm going to take my funnel out, put that off to the side, and now I'm left with a juice. And this juice still does have some soap in it. So I'm going to use the ethyl alcohol to bring the DNA to the surface and then use my wooden skewer to scrape it up and pick up the DNA. So I'm going to hold my beaker at an angle and very carefully pour my ethyl alcohol over the top. Now this just came out of the freezer, so it is nice and cold in comparison to the rest of our mixture. Now, very carefully, I'm going to scrape the top, not stir, but scrape the top. And what my wooden toothpick is going to do is actually pick up my DNA. So that right there is the accumulation of the strawberry DNA. Now, if I continue to be really careful, I might be able to pick up some more. So that is the Strawberry DNA Lab.